Hello, everyone. My name is Francis Chikweto. I'm coming from Zambia, Evelyn Hon College, and I'll be presenting my work, which we are doing with my friend Jordan from the University of Zambia, which is focused on diagnosis of cervical cancer using convolutional neural networks, deep learning. Cervical cancer is the fourth leading cancer in women. And last year, there was about 570 cases that were reported. And 90% of such cases actually occur in countries like Zambia, which are developing countries. And currently, my country is amongst the leading countries in cancer cases, cervical. And this is why we took this as a challenge, where we can use deep learning actually to try and see if it can be used for screening of cervical cancer. The reason why we have such cases that in my country, even in other developing countries, we do not have enough trained pathologists, and also the laboratories that we have are kind of limited in terms of technology. So we tried to come up with a screening tool that can be fast enough to help the fewer pathologists that we have to take decisions. So for the data sets that we used the foreign data set from the National Cancer Institute, which is, in this case, it's open access, and it has slide images, high resolution. And also, we also used our own local data set from the University Teaching Hospital, the main lab. And we collected a sample of cancerous and also normal tissue slides, and the number you can see it on the screen. The challenge we had is that these slides are SVS files, and they are so large. So the first thing that we had to do was actually to reduce the size of the images that we have so that our model can easily read. And therefore, from the high resolution SVS formats that we had, the slides, we generated the 2034, 1356 pixel images, and those are the ones that we used now actually to crop them further to a smaller size, 256 by 256, and from there we did the augmentation of data so that we generate more data for our model. And the model was now ready. The images now are ready to be fed into the light DNA architecture that we used. For the results, for the best model that we came up with so far, we compared it with the VGG, which we used transfer learning to train it, and then we compared the validation data. If you look at the results, we found that VGG it even took long to train, but our light model actually performed quite all right on this data set. Then for the other results, which is the confusion matrix, we wanted to see if the foreign data that we got from the National Research Institute can actually predict the cancer in the cells from our own hospital. And so the confusion matrix now, there you are able to see the results. We used the local data sets now to test our model from the University Teaching Hospital. And we found out that it was able to predict up to some accuracy. Yes, this is research is not yet complete, it's ongoing. And from the results that we have gained so far, it has potential actually to improve the cancer cases and screening processes in our country. And we are hoping that in the future, through this conference and the lectures that we are receiving a lot, we can actually come up with better models and also the architectures to help us to improve this model that we have developed. So thank you so much. We'll see you at the poster.